Moving forward with chemistry of carbohydrates, third lecture. So the specific learning objectives for today's lecture are we'll classify polysaccharides with examples, describe homopolysaccharides like starch and glycogen as storage form, and we'll also look into what are cellulose, inulin, and dextran. And lastly, we'll describe what are heteropolysaccharides like glycosaminoglycans and what is the role as structural elements in the human body. Okay, so let's move forward. So polysaccharides are basically classified as homopolysaccharides or heteropolysaccharides. Okay, so homopolysaccharides are basically more than 10 sugars which are joined together by glycosidic bonds. Okay, and all these 10 sugar and more than 10 sugars are same sugar units. Okay, so there is no difference in sugar units. Whereas in heteropolysaccharides, it has more than 10 sugar units which are joined together, but it contains different sugar units. Okay, so the term homo means same sugar units and heteropolysaccharide means different sugar units. So the examples of homopolysaccharides are starch, which is made up of only glucose units. It is seen in plants. Glycogen, which is again made up of only glucose units and is seen in human beings and animals. Cellulose, which is again made up of only glucose units, seen in plants. Inulin is made up of fructose units. Dextran is made up of glucose units, and chitin, which is skeletal of the insects, is made up of N-acetyl glucosamine. Okay. Now if you look at heteropolysaccharides again, the simplest example is agar, which is made up of glucose and galactose, and it's a nutrient medium for growth of organisms in case of for investigation purposes. Okay. The next we have is hyaluronic acid, heparin, chondroitin sulfate, keratin sulfate, and dermatan sulfate. So these are all the examples of glycosaminoglycans or mucopolysaccharides, which we will be seeing later. Okay, so the examples of heteropolysaccharides, which contain 10 or more sugars, more than 10 sugar units, they contain different sugars, for example glucose and galactose. So the agar is the simplest form of heteropolysaccharides, and these hyaluronic acid, 2-dermatan sulfate, these are all mucopolysaccharides or glycosaminoglycans. So first let's look at starch. What is starch? A starch is basically a homopolysaccharide of glucose units. So it is made up of only glucose. It is the storage form of sugar in plants, the storage form of glucose in plants. Okay, it's a reserve form of glucose in plants. The sources of starch is potatoes, tapioca, cereals, etc., okay, which we consume in our diet. So these things which we consume in a diet, potatoes, tapioca, cereals, they are a rich source of starch. Next, starch while boiling forms two parts. One is a soluble part and the other is an insoluble part. So the soluble part is roughly less, which contains about 20%, and it is known as amylose, and it's made up of 1,4 bond, whereas the insoluble part is the major chunk, which is 80% approximately. It is called amylopectin and it is 1,6 bond. So if we look at the structure of amylose, so which contains glucose unit and you have a 1,4 bond. Okay, again we have n numbers of amylose, n number of glucose units. So each glucose unit is having a bond of 1 and 4, the first carbon atom and the fourth carbon atom. If you look at amylopectin, so amylopectin has a branch. Okay, so it has a 1 and 6 bond, which forms the branch. So amylopectin is again made up of only glucose. Okay, so the main chain might have a 1,4 bond, and the branch chain which is there, it has a 1,6 bond. Okay, and the branch again, it may have a 1,4 bond. Okay, so amylopectin is the branched form of homopolysaccharide, okay, which is the insoluble starch, and it is made up of 1,6 bond, okay, at branching points. Now starch is hydrolyzed by the enzyme which is known as amylase. So in our body, when we take in starch, it is hydrolyzed by the enzyme which is called amylase. Now we have two types of amylase. One is present in the salivary amylase and pancreatic amylase based on the location. So both salivary and pancreatic amylase, they act on 1,4 bond. Okay, so the amylase will act on 1,4 bond and eventually lead to breakdown of starch and form is maltose and dextrin. Okay, the relatively bigger part of hydrolysis of starch is called as dextrin, and the smallest part to which the amylose can reduce the starch to is called maltose. Okay. Starch again has a high glycemic index, so it is best to avoid starch for diabetic patients. Okay, so why starch has a high glycemic index? Starch has a high glycemic index because it is made up of glucose units only. Okay, so it has high glucose units, so the glycemic index, or the ability of the food to increase the blood sugar level as compared to the reference glucose, is high. Okay, so starch will increase more blood glucose levels, so that is why it has a high glycemic index, and it is best avoided for diabetics. Starch is again a non-reducing homopolysaccharide. Okay, it has very few functional groups, so that is why it is considered as non-reducing. Okay. Now we have a test which is specific for starch, which is iodine specific test, in which the starch, when reacts with iodine, it forms a blue color. Okay, so this is about starch, which is made up of glucose subunits. Okay, so starch on boiling will form two parts, which is a soluble part and an insoluble part. The soluble part is called amylose and it has a 1,4 bond only, whereas the insoluble part is called amylopectin, which has 1,6 bond at various branching points. Okay, so in our body we digest only amylose because we have the enzyme amylase for it. So the amylase acts on amylose, whereas we do not digest amylopectin, okay, because we do not have the enzymes for it. Next is glycogen. So glycogen again is a homopolysaccharide which is made up of glucose units. Now glycogen is the storage form of glucose in human beings as well as in animals. Okay, it is present in muscle and it is present in liver. The glycogen which is present in the muscle is used to provide energy which is required by the muscle, whereas the glycogen which is present in the liver, it maintains the blood glucose levels. So whenever the blood glucose levels goes down, so that is again brought up or it is maintained by the glycogen which is present in the liver. So the glycogen again, it has 1,4 and 1,6 glycosidic bond, and it is highly branched molecule. So you can see over here, this is the glycogen structure. Okay, it has high branching points. So if you look at the main, this is the main stem. So each dot represents a glucose molecule. So each dot has a 1,4 glycosidic bond. Okay, glucose is attached as 1,4 glycosidic bond, and these branching points, at this branching point it has 1,6 bond, just like it was there in starch. Okay, so it has high branching points. You can see lots of branches are here, so it's a highly branched molecule. Okay. See, defect in glycogen is associated with glycogen storage disease. Okay, it's basically an inherited disorder in which glycogen is either not utilized or the glycogen which is deposited is an abnormal glycogen. Coming to other polysaccharides, for example, cellulose, dextran, and inulin. Cellulose again is made up of glucose units. It is found in plants, and it has a beta 1,4 glycosidic bond. Okay. See, if you look at the other storage form of carbohydrate in plants, which was starch. Okay, so starch was alpha 1,4 glycosidic bond. So here we have is a beta 1,4 glycosidic bond. Okay, so we do not digest it, we cannot digest this because we do not have the enzymes to digest the cellulose. So even because of the orientation of the anomeric carbon atom, okay, just because the orientation of hydroxy group is changed, okay, we have is beta 1,4 glycosidic bond, and we do not have the enzymes which can digest this. So just by change in the orientation of hydroxy group, okay, the enzymes are not able to act on this. Okay. So this is a beta 1,4 the glucose units are joined together by beta 1,4 glycosidic bonds, and we do not have the enzyme to digest this bond. Okay, so the enzyme which is required to digest this bond is called cellobiase, which is present in animals and not in us. Okay, so it's absent in humans. However, even though we do not digest it, so this becomes an undigested carbohydrate which forms the dietary fiber. Okay, so this constitutes dietary fiber. Again, dietary fiber has various health benefits. It helps. It is used to lower the cholesterol in our body. It is also beneficial for constipations. Okay, it provides roughish to the food, which helps in constipation, and it is also believed to be play some role in various cancer disorders. So this is an important component of dietary fiber. Next is dextran. Dextran again is made up of glucose. Dextran is produced by microorganisms, and again it is a highly branched molecule and it is high molecular weight. Okay, so it has 1,4, 1,3 and 1,6 glycosidic bonds. It has a high molecular weight. So, and because of this high molecular weight, when we give it as in an intravenous solution, intravenous injection, so it stays in the plasma and it is not able to move out of the plasma. So this increases the plasma volume. Okay, so we can use this in case if there is loss of plasma volume because of a sudden loss of plasma volume because of dehydration, or let's say road traffic accident, increased blood loss has occurred. Okay, so by the time we arrange for blood, we can give this dextran as a plasma volume expander, and which maintains the normal blood pressure. Next we have is inulin, which is made up of fructose. So inulin is present in onions, garlic, tubers, etc. This again has a beta 1,2 glycosidic bond. Okay, because it's made up of fructose, so it has a 1,2 glycosidic bond. Again, it is not metabolized because we do not have the enzymes to metabolize inulin. And inulin is used to measure the GFR, which is glomerular filtration rate, which is again a function of the kidney. So it's basically inulin is used to assess how well the kidneys are functioning. Okay, so we measure the GFR, which in turn measures the function of the kidney. Coming to heteropolysaccharides or glycosaminoglycans. So heteropolysaccharides are also called mucopolysaccharides. Okay, so they are made up of repeating units of uronic acid and amino sugar. Now, what is the uronic acid? Now, if you look at this glucose structure, okay, the functional group is oxidized to carboxylic acid. Okay, here is it was a functional group at carbon atom C1. Now, if this functional group is oxidized to carboxylic acid, it forms as gluconic acid. However, if the functional group is spared and the last carbon atom is oxidized to carboxylic acid, it forms as glucuronic acid or a uronic acid. Okay, so this is glucose, so it forms a glucuronic acid. So this is an example of uronic acid. So uronic acids are formed when the oxidation occurs at the last carbon atom, which forms the carboxylic group, and the functional carbon atom is spared. So oxidation is spared over here. So it is the repeating unit of uronic acid and amino sugar. Okay, so glycosaminoglycans, okay, it's a heteropolysaccharide which contains repeating units of uronic acid and amino sugar. So amino sugar is when the amino group is attached to sugar molecule. So we have as repeating units of uronic acid, amino sugar, uronic acid, amino sugar. So that repeating unit forms a glycosaminoglycans. So these glycosaminoglycans are negatively charged because the hydrogen atom is lost, so it becomes COO minus. So these glycosaminoglycans are negatively charged, and they attract water molecules and they make the solution viscous and has a cushioning effect. Now you can see over here in this structure, you can see H2O water molecules which are attached with these red strands are glycosaminoglycans. So because glycosaminoglycans are negatively charged, they attract water molecules. Okay, so when let's say there is a compression, so the glycosaminoglycans, the water molecules, they come out. Okay, and this glycosaminoglycans which is negatively charged, and let's say we have another glycosaminoglycans GAGs over here which is also negatively charged. So this negatively charged and this negatively charged, they repel each other. So they have an outward force which is developed along with water. So when we have compression, so this force is again counteracted by the outward force of the water as well as the because of charge of the glycosaminoglycans. Okay, and when the compression is removed, again the water molecules, they go inside and assume the resting state. So this is how the glycosaminoglycans, they also form a cushioning effect, okay, and they make the solution viscous. Okay, so they have tags, they repel each other and they form a resilient elastic structure. Now, what is proteoglycan? So, okay, proteoglycan basically is a protein core having glycosaminoglycans which are covalently attached. Okay, if see over here, so this is a proteoglycan. Okay, so these yellow dots, this is a protein or polypeptide backbone. Okay, so this polypeptide backbone, these green dots are glycosaminoglycans. The polypeptide backbone, if glycosaminoglycans are attached, it is called proteoglycans. Okay, so this you can have many proteoglycans which are linked to a hyaluronic acid, which is also a proteoglycan. Okay, and what is glycoprotein? So glycoprotein is similar to proteoglycan. The only difference is glycoprotein also has a polypeptide backbone, but to this you have is oligosaccharide chain which are covalently attached. So in proteoglycans you have glycosaminoglycans which are covalently attached, whereas in case of glycoprotein you have is oligosaccharide which are covalently attached. Now if the carbohydrate content is more than 10%, then that glycoprotein is called mucoprotein. Okay. Now this is another structure of proteoglycan, okay, which has a polypeptide core to which the glycosaminoglycans are attached. You can see over here it has one collagen type fiber which is here and this another collagen fibers which is here. So the proteoglycan is holding together the collagen fibers. So it provides this structure, maintains the structural integrity of the cell, okay, or structural integrity of the tissue. Okay, so this is again important function of glycosaminoglycans. Now the important glycosaminoglycans are, the first one is hyaluronic acid, which is made up of N-acetyl glucosamine and glucuronic acid. So as we discussed earlier, the glycosaminoglycans are repeating units of uronic acid and amino sugar. So the uronic acid here is glucuronic acid and the amino sugar is N-acetyl glucosamine. Okay, so glucose to which amino group is attached to it is glucosamine, and to also the acetyl group is attached, so it becomes N-acetyl glucosamine. So it is present in soluble fluid, vitreous humor, and it acts as a lubricant. Next is heparin, which is made up of again glucosamine and iduronic acid. Now iduronic acid is basically, it's a uronic acid of idose sugar. Idose is again an epimer of glucose. Okay, so idose sugar, it forms as iduronic acid. So iduronic acid and glucosamine. Heparin is present in blood, lungs, kidneys, spleen, and basically is an anticoagulant. Next we have is chondroitin sulfate, which is made up of N-acetyl galactosamine and glucuronic acid, and it is present in cartilage, bone, skin, blood vessels, and it maintains the structure of all these. Next we have is keratin sulfate, which is made up of N-acetyl glucosamine sulfate and galactose. It is present in cornea and cartilage, and in cornea it is one of the ways by which the cornea is transparent. It keeps the cornea transparent. Okay, if the cornea is not transparent, then we will go blind. Next is dermatin sulfate, which is again made up of N-acetyl galactosamine sulfate and iduronic acid. Okay, and it maintains maintains the various structures. So these are the important glycosaminoglycans, or biomedical importance of glycosaminoglycans. Okay. So to summarize, we talked about polysaccharide classification. So polysaccharides are again homopolysaccharides or heteropolysaccharides, which contain more than 10 sugar units which are joined together by glycosidic bond. Now if all the 10 sugar units are same, it becomes homopolysaccharide. If it contains different sugar units, it is heteropolysaccharide. Okay, so example of homopolysaccharide is again starch, glycogen. Okay. Whereas heteropolysaccharide we have is agar and glycosaminoglycans. Starch again is made up, it's a reserve form of carbohydrate in case of plants. It is made up of repeating units of glucose. Like it has one 4 bond and one 6 bond. Okay, starch on boiling or cooking forms two parts, there is a soluble part and an insoluble part. So the soluble part is called amylose, okay, which has only one 4 bond, and we can digest it so by the enzyme amylase, which is present in saliva and the pancreatic juice. Okay. Whereas the insoluble part, which is formed by the cooking of or boiling of starch, is amylopectin, which has a 1,6 bond. Okay, and the product of breakdown of starch by amylose is maltose and a relatively bigger molecule which is called dextrin. Then we talked about glycogen. Okay, so glycogen again is a reserve form of a storage form of carbohydrate in case of human beings and animals. So it is made up of glucose. It is highly branched. Okay, and it is present in muscles and liver. So in muscles, the function of glycogen is to provide energy, whereas in case of liver, it maintains the blood glucose level. And then lastly, we discussed about various glycosaminoglycans, which are again repeating units of uronic acid and amino sugar. Okay, so thank you very much and see you next time.